Hello, everyone, and welcome to today's live webinar, Using Reference Standards to Validate Your NGS Oncology Workflow, presented by Hannah Child, Diagnostics Product Manager at Horizon Discovery. And I'm Whitney Ghaffari of Horizon Discovery, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Horizon Discovery. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you would like at any time during this presentation. To do so, simply type them into the Ask a Question box located on the far left of your screen and click on the Send button. We'll save several questions for Hannah to answer during the Q&A session. Additionally, we have scientists standing by to answer questions as they come up during the presentation. We'll answer as many questions as we can. If there are questions that we don't get to, we'll send an email response in the coming days. Additionally, when this presentation is available for on-demand viewing, questions can still be submitted, and they'll be routed to Hannah and the research team for an email response. Also, please notice that you'll be viewing this presentation in the slide window. To enlarge this window, click on the two-arrow symbol at the top right-hand corner of the slide. If you have trouble seeing or hearing this presentation, click on the Support tab found at the top right of the presentation window, or report your problem through the Ask a Question box. Continuing education credits are offered for attending this webinar. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. Also, check out the other tabs that contain helpful resources that may answer questions that will come up during this webinar. I'd like to now introduce our presenter, Hannah Child, who will be taking us through the tips and tricks learned through her years of researching reference standards. Hannah joined Horizon as a product development manager in 2016, working with the scientific research and development team to develop new reference standards before transitioning to the commercial team as the Diagnostics Product Manager in 2017. Prior to joining Horizon, Hannah was project manager for the Stratified Medicine Scotland Innovation Centre, SMS-IC, in Glasgow, where she supported the establishment of a new 8-sequencer Ion Proton NGS lab and data center in collaboration with the Thermo Fisher Scientific. After completing her PhD in molecular cell biology at Glasgow University, Hannah worked as an analyst and then as a study manager at BioOutsource, a Scottish CRO specialising in immunotherapy screening assays for biosimilar antibody efficiency testing. Hannah holds a PRINCE2 Practitioner Certificate in Project Management and obtained her BSc Hans degree in molecular biology from Edinburgh University. Now for the presentation. Over to you, Hannah.
Thank you, Whitney. We have 3 main learning objectives for the webinar today. Firstly, we hope to help you learn some of the main criteria to check when introducing a new NGS assay into your lab's testing menu. This includes criteria such as looking at your accuracy and specificity, the assay's limit of detection, and how you may handle challenging to sequence variants, like structural variants. Secondly, we hope you'll feel more confident to recognise how to select the most appropriate reference standard for your assay, taking into consideration the allele frequency, the range of variant types you should include, the gene panel coverage, and the DNA format that will be most commutable to your real samples. Finally, we hope to go through the best practices for initial assay validation and routine quality monitoring, including key features such as robustness of your assay, user competency, and how you may handle your change control procedures. The majority of the reference standards that we manufacture at Horizon are aimed at the NGS user group. NGS stands for Next Generation Sequencing. And the two main providers of NGS platforms are Illumina and Thermo Fisher Scientific, both companies offering multiple different sequencing models. Here you can see them displayed on the slide. Both technologies offer sequencing by synthesis, with Illumina's technology being based on fluorescence and the Thermo Fisher Scientific Ion Proton technology being based on charge. Irrespective of whichever sequencing platform your lab may be using, the same basic workflow applies to the majority of NGS clinical assays, and errors can be introduced at any stage of the workflow. This includes some of the early steps, such as extracting samples from the patient, whether that be a tissue biopsy set in FFPE, or a blood sample perhaps from liquid biopsy. How different labs handle the samples and how they are processed in the early stages can be very critical, and we see handling errors introduced at this early stage. Next, you have extraction of your analyte from the original sample, whether your analyte be RNA or more commonly DNA. And following the extraction, you will want to quantify and qualify your analyte. And there are many opportunities for error to creep in at this early stage. Following initial sample analysis, the analyte is prepped for the assay, which in the terms of NGS normally includes library preparation and templating, and finally put onto one of the sequencing workflows. The last step in the process is the bioinformatics analysis, and this can often be the most complicated and error-prone in the entire process, with many different settings and optimization options for labs to tackle. And it's very important to use reference standards to make sure that this workflow is all functioning correctly. The end goal is to be able to get the correct test result back to the patient. To go into the problems around NGS in a little more detail, as mentioned, the ideal final output of NGS assays is a report listing the true variants that are found in a clinical sample. However, due to the following technical issues, this can often be problematic. For example, the sequencing platform itself can have difficulties in physically incorporating the correct base. This can be due to the natural error rate of DNA polymerase, or perhaps amplicon dropout due to poor primer design of the gene panel. Some DNA templates can also be more difficult to sequence, and this includes CG-rich regions or perhaps hairpin loops, which can inhibit the activity of DNA polymerase and possibly have unusual melting temperatures which can affect sample preparation. Additionally, homopolymers can be difficult for some platforms, such as the Ion Proton platform, which finds it difficult to differentiate between an accumulation of charges in long stretches of homopolymers. We see the majority of clinical samples coming from FFPE, which is formalin-fixed, paraffin-embedded samples. This is because this type of sample preservation technique was historically used for microscopy, but due to the high abundance of samples, we now see this as the main input into our clinical NGS labs. While good for microscopy, the FFPE process causes damage to the DNA that is very difficult for NGS to handle. In particular, it can cause deamination of the DNA, where C and T can be mutated into TNA bases. And this can cause artificial false positive mutations to be called in your final results, which may not be a true result of your patient's genetics. You can see on the right there a paper I've highlighted describing this issue of deamination in more detail. Finally, in relation to the bioinformatics pipeline, alignment of NGS data to a reference genome can cause multiple headaches for the analysis team. Questions can be asked around which is the most appropriate reference genome to use, and interestingly, we see the field possibly moving towards more intelligent graph genomes than linear, as are currently in use. We see people becoming more interested in sequencing tumor-normal matched pairs to identify somatic mutations in cancer, and also in sequencing parent-offspring trios to identify novel germline mutations where these samples are available. The solution to controlling your error rate in NGS is to use reference standards to monitor every step of the process. A molecular reference standard is a defined substance, either DNA, RNA, or protein, which is used as a measurement base or control for an assay. A reference standard should have a number of key features which are essential to its use. It should be homogeneous and stable with respect to its specified properties. And these properties can be qualitative, such as the absence or presence of a particular variant, and quantitative, such as the abundance of this variant within the sample. The reference standard should be consistent and reproducible between batches, and possibly most importantly, it should be commutable, which means that it should closely mimic real clinical samples. Finally, reference standards should be characterized using a number of different procedures or by a procedure considered to be the gold standard. And at Horizon, we predominantly use digital PCR. Horizon's reference standards have a number of key features which makes them a good choice for NGS labs to use. They are cell line derived, which means that they closely mimic the patient genetics and may be more appropriate for use as a reference standard than perhaps some synthetic plasmid alternatives. This means that the variants are presented in the relevant genomic complexity. We have multiple sample formats available, mimicking real samples, such as FFPE, RNA, or formalin-compromised DNA. And our reference standards are available at a range of allelic frequencies to best match your needs. Our controls are produced in a quality-controlled and validated environment and prepared under a certified quality management system. And we have both ISO 9001 and ISO 13485 in place in our manufacturing facility in the UK. Horizon's unique approach to manufacturing reference standards stems from our expertise in gene editing technology. As this schematic outlines, we begin with a wild-type cell line. We single-cell dilute that cell line to produce a clonal population. We then can perform precision gene editing to create the clinically relevant mutations that our customers require and validate the final mutant cell line. Based on this technology, we have a wide range of materials available. This slide highlights some of the main categories we have. In particular, it's useful to point out the cell-free DNA, which is the reference standard you would want to use in the growing field of liquid biopsy. We also have multi-gene multiplexes for larger gene panel use, and our largest reference standard is called OncoSpan, which covers 152 cancer genes. We have single gene multiplexes if you're running a particular BRCA, EGFR, or KRAS gene panel. RNA fusion reference standards, base-seq, genome in a bottle in FFPE, and finally we have a very experienced custom design team who can help you if you need something a little more bespoke. Horizon's reference standards can be inserted into an NGS workflow at two different points. We have multiple formats available to be incorporated at the earlier stages of the workflow to test the DNA extraction procedure. And this includes our plasma cell-free DNA standards and our FFPE blocks. Alternatively, our cell-free DNA and buffer, genomic DNA and formalin-compromised DNA products can be inserted directly into the library preparation and should be handled in a similar way to your other real samples. In this way, reference standards are suitable for three main applications. We see a lot of our customers using them for assay development to guide them as they may be creating new gene panels, assay validation when people are bringing in a new gene panel into their testing menu, and routine monitoring, where labs are continually using reference standards on every run to provide evidence and audit that their quality procedures are up to scratch. Some of the main criteria to check when introducing a new assay into your lab's testing menu are outlined in the 7 points below. It's important to check the accuracy and the specificity, looking at are the variants detected that are not present, and these are termed false positives. What's the sensitivity of the assay? Can the variants be detected if they are present? What's the limit of detection? What is the minimum allele frequency that can be detected? And how does the assay handle structural variants? How do these difficult to sequence variants perform on your workflow? Additionally, what is the repeatability? Is there any run-to-run variability? And reproducibility. Can the results be reproduced at perhaps different sites? If you might have a UK and a US site, it's interesting and important to compare performance. Finally, it's important to check the robustness of your assay. What is the effect of sample quality on performance? When using a reference standard to control for error, labs require a sample with known sequence, and this can be termed in the field as a truth set. Looking at accuracy, specificity, and sensitivity in a little more detail, it's important to test the ability to identify the correct variants that are truly present within a sample, whilst minimizing the level of both false positives and false negatives. False positives being the case that you call a variant that is not actually there, and false negatives being the case when you do not call a variant that is actually there. And both of these outcomes can be very impactful to real patient testing if they were to occur on a real sample. Looking at sensitivity, it's critical to identify the assay's limit of detection. This is because cancer samples are heterogeneous in nature and have a mixed normal and tumour cell population within one sample. And somatic variants that may be driving that cancer could only be present in a subpopulation of the tumour cells. This means that labs want to be able to know how low they can accurately call variants. And we see our clinical labs looking at around 5% allele frequency. If labs can call the reference standard accurately at, for example, 2% variant allele frequency, then it gives them confidence that they can accurately call rare variants in real samples as low as 2% variant allele frequency. As mentioned, it's also becoming increasingly important to understand how your assay handles difficult to sequence variants. This is becoming of increasing importance as we gain knowledge about more and more structural variants being involved in driving cancer, and more of those structural variants are becoming actionable with associated therapies. Therefore, if clinical oncology samples are likely to contain these variants, it is important to validate the workflow with reference standards that also contain them. The types of variants that we're talking about here are structural variants, and some of the main types have been listed below. This includes insertions and deletions, often termed indels, copy number variants, termed CNVs or gene amplifications, gene fusions, homopolymers, which are stretches of repetitive sequence, which can be challenging for some platforms, and high-low GC regions. Interestingly, clinical samples may often contain perhaps only one or two of these structural variants at a very highly characterized level. However, one of the main advantages of using commercial reference standards is that they have the ability to contain multiple variants at once. So you'll be saving yourself time and money because you'll only need to validate your assay with one set of material. Moving on to look at how you may select the most appropriate reference standard for your assay. We're going to touch on four main points: the variant allele frequency, the range of variant types, the gene panel coverage, and the DNA format. It's important to select a reference standard that contains variants present at variant allele frequencies, or VAR, close to the intended limit of detection of your clinical assay. This table shows some of the main multiplexes that we provide here at Horizon, and the number of variants that those products contain within the three different bands outlined in the table. So to start on the top row, our largest reference standard, OncoSpan, contains 13 variants at between 1 to 10 allele frequency, over 300 variants between 10 and 50%, and 35 variants between 50 and 100%. Alternatively, our structural multiplex reference standard contains 7 variants, all within the 1 to 10% bracket. Our quantitative multiplex standard contains variants at both the 1 to 10 and the 10 to 50% brackets, as does our BRCA somatic multiplex 1. To point out our true Q7 product here, we have 40 variants present at 1.3% variant allele frequency. So this product would be very good for labs who are really wanting to challenge themselves and see if they can get down to that very low level of allele frequency. Fundamentally, a tumour is a mass of cancerous and non-cancerous cells, and so therefore, as mentioned, the cancer load is expressed as a percentage, the allele frequency. Horizon reference standards are able to mimic tumours by combining normal and mutant cells together to achieve the ratios outlined in the previous products. So we can really create the allele frequencies that are required. This slide shows an example dataset of how our multiplex 1 cell-free DNA reference standard set may be used to help calculate the limit of detection and false positive error rates of an assay. You can see here that the reference standard contains 8 different variants, and the product is available in 4 different tubes, with all the variants being present at 5%, 1%, 0.1%, and wild type. In this example dataset, you can see that the lab was able to correctly call all 8 variants at the 1% allele frequency, but was not able to call them at the 0.1%. This informs the lab that their correct limit of detection can be considered as 1% allele frequency. Interestingly, this dataset also points out that the lab may be having problems with the NRAS A59T mutation, and they are possibly calling a false positive here, since they had a positive reading in the wild-type sample. This type of use of reference standards allows pipeline optimization and provides confidence in results when handling real patient samples. To look at the second criteria, it's important to check the range of variant types in the reference standard. And it's good to select a reference standard that contains variant types that are likely to appear in your clinical samples. Our structural multiplex reference standard contains a range of different mutation types. So we have copy number variants, fusions, indels, and variants present in high-low GC regions. You can see that the structural multiplex reference standard is available in 3 different formats to best meet your needs: FFPE, genomic DNA, and cell-free DNA. Additionally, our largest reference standard, OncoSpan, contains 30 indels. And in 2019, we're hoping to add more structural variants to this large product, including copy number variants. We also have a range of RNA fusion reference standards, which are available in FFPE. These contain 5 clinically relevant RNA fusions, as described in the table below, in ALK, RET, ROS1, NTRK1, and NTRK3. It's useful that this product is also available with a matched negative control. And you'll see on the right there that we had a very nice white paper published in conjunction with Thermo Fisher Scientific last year, where our RNA fusion reference standards were recommended for use alongside their Ion Amplific and Oncomine, ALK, RET, and ROS1 fusion assays. And you can read more about that white paper on our website. Thirdly, looking at gene panel coverage. You should select a reference standard that contains variants that will validate a large proportion of your gene panel. And we see our user base moving towards larger and larger gene panels as more variants become actionable. For this reason, last year we launched our OncoSpan reference standard, which contains 386 variants across 152 oncorelevant genes, and this product has been validated by exome sequencing. In the table there, you can see the gene list of the 152 genes that are covered in this product. And this slide outlines how OncoSpan can be used across different gene panels. So we've done the comparison to see what the gene coverage is like on the main commercial gene panels in use. So to point out one aspect in particular, you'll see on the Illumina TrueSight Tumor 170 gene panel, OncoSpan reference standard covers 74 genes within that panel. This allows wider panel validation than is possible with most clinical samples, which may only contain a couple of variants in a handful of genes. By using OncoSpan, you're able to validate a much larger proportion of your gene panel in a more efficient manner. Finally, it's important to look at the DNA format of your reference standard and to select a reference standard that is the most commutable format to your real clinical samples. At a high level, all of the Horizon reference standards are cell line derived, which means that they're more similar to real samples than some synthetic plasmid alternatives. At the next level down, all of our cell line derived reference standards are available in multiple formats, So we have cell-free DNA reference standards available for liquid biopsy, and the format of these standards is fragmented DNA, with the average fragment length of 160 to 170 base pairs. In addition, last year we launched some of our cell-free DNA reference standards in synthetic plasma, which allows labs to validate the early DNA extraction step. We also have FFPE curls for labs who are handling DNA from FFPE samples, and formalin-compromised DNA reference standards, where the DNA has been treated with formalin for different time lengths to produce either mild, moderate, or severe DNA damage with decreasing DINs, as tested on the tape station. The formalin-compromised DNA reference standards would probably be of most use to labs who may be handling damaged DNA, but may not actually have extraction facilities themselves, whereas labs who are handling the FFPE curls would want to opt for an FFPE reference standard. You can see here in the schematic some of the main validation steps that we do for our FFPE-based standards are gel electrophoresis and analysis on the tape station. Related to the topic of DNA format, this slide outlines some of the benefits of using our synthetic plasma control. We see many disadvantages when using real human plasma, such as variable quantity and concentrations, lot-to-lot variability, irregular supply, contamination with other analytes or genomic DNA, and cell-free DNA degradation, where real samples have time-limited storage. As a solution to these problems, we have found a synthetic plasma to be used as a matrix for our cell-free DNA reference standards. The advantages of this matrix are that we have defined volume and concentrations, controlled lot-to-lot stability, a reliable supply, no interfering analytes or genomic DNA, and long-term stability of cell-free DNA beyond 24 months, which is very important for reference standards as they may be getting used over long time periods across multiple labs in controlled studies. Here we have some data from one of our studies which shows the performance of the synthetic plasma compared to human plasma. And this data set really gives confidence that the synthetic plasma matrix mimics human plasma in terms of how much cell-free DNA we were able to recover during extraction and in the detection of gene copy number variants. The next part of the webinar will touch on some best practices for the NGS community. Some of the key features here we're going to discuss are robustness, user competency, and change control. We often get our customers coming to us to ask about best practices and how they should be using the reference standard. So it's really good to share some of the tips and tricks and expertise that we've amassed along the way. We find that using reference standards supports our customers in their own quality audits that they may be going through to achieve their ISO or their CLIA lab accreditation. And some of the important points to focus on are during initial assay validation, it's critical to check assay robustness on different days with different sample qualities and on different sites, if applicable. It's also important to assess user competency, and reference standards can be used to ensure consistent accuracy of results across multiple users. This can also be very beneficial to labs who might have new staff to train and to prove their competency during audit, where the results of how they sequence the reference standard can be kept in their training file. Finally, it's very important to monitor change control. We see this something We see this as one of the main features that our customers need support with, where the best practice is to run the reference standard on every sequencing run. This helps labs to monitor for change in accuracy over time. And some case studies that we've seen crop up are that errors can be introduced when new reagents are brought into a workflow, and perhaps to qualify new version releases of analysis software. So for some of the bioinformatics software, our customers may be handling multiple version releases per year, and this can be a dangerous area of error introduction. So running a reference standard on every run allows them to flag up and notice any change in accuracy with these change in reagents on software. Finally, labs are often subject to changes in location, where equipment needs to be moved between labs or even to new buildings. And the recalibration step to make sure the equipment is performing as it was before is essential. And running reference standards before and after the move can help to confirm this. Finally, I've included some screenshots of some of the best guidelines that are currently available for NGS labs. Here we can see an article that was published in 2017 called Guidelines for Validation of Next-Generation Sequencing-Based Oncology Panels. And this is some of the latest recommendations from the College of American Pathologists. Secondly, we see a lot of our users relying upon the New York State guidelines. And here we have a publication from them in 2016 called Next Generation Sequencing Guidelines for Somatic Genetic Variant Detection. And this remains one of the gold standard guidelines in place today for NGS labs. Finally, there was an interesting paper that came out last year in Nature Reviews called Reference Standards for Next Generation Sequencing by Hardwick et al. And as highlighted in the abstract here, they concluded that the errors associated with NGS can be understood and mitigated through the use of reference standards. So to finish, I've included some links to helpful resources here. The top link is a link to our website for more information on the reference standards described in this presentation. We also have an excellent app note section where you can read more about technical information. Finally, I've put in links there to the 3 guidelines that were described in this presentation: the CAP guidelines, New York State guidelines, and the Nature Review paper. I'd like to thank you for your time and attention and pass over to Whitney to handle any questions.
Wow, thank you so much, Hannah, for this informative presentation. We'll now start the live Q&A portion of the webinar. Now, while we're getting ready for the Q&A session, 2 polling questions will appear on your screen. We really appreciate your answers to these questions so we can follow up with you appropriately. I'd like to remind our audience that you can submit questions by typing them into the Ask a Question box located on the far left of your screen. Just type your question into the box and hit send. We'll answer as many of your questions as we have time for. Okay, Hannah, are you ready for some questions?
Yep, I'm ready.
Awesome. Okay, so the first question in the queue is, what are the advantages of clinical oncology testing by liquid biopsy as opposed to from SSPE?
Okay, thank you. That's a good question. I think one of the main advantages we're seeing in the field is that it really holds a lot of promise to make testing more accessible and easier for the patients, which is hugely important. So when clinicians are obtaining samples to be used in the FFPE workflow, the patient has to go through a surgical procedure to obtain that biopsy sample, whereas the new field of liquid biopsy allows the DNA to be collected from a simple blood draw from the arm. So it's much less invasive for the patient, and also it means that due to the increased accessibility of DNA, it opens up new doors to routine monitoring and increased testing for the patient over time, perhaps to look at changes to therapy response, and also to monitor how their cancer may progress long term.
That does seem like that makes a big difference. Wow. Okay. Great. I have another question here. The second question is, do you offer Conospan in any other format?
Okay, currently our Oncospan product, which we launched last year, was launched in gDNA only. However, we have had multiple custom requests to provide this large reference standard in cell-free DNA format. So that's definitely something that is available through our custom team at the moment. And we are launching Oncospan in FFPE next year, and it's most likely that cell-free DNA version will follow due to the high demand.
Wow. Okay, let's see. Oh, here's another one. Okay, so this question is, they want to know, Hannah, is it possible to make a bespoke blend, and I'm not even sure if I'm pronouncing that word right, a bespoke blend if we need a different allele frequency?
A bespoke blend? Yeah, so in our off-the-shelf catalogue, we have over 100 reference standards with the allele frequencies determined and qualified for every batch, and many we touched upon today. However, some of our customers may need something a little different, and if you require an allele frequency or even additional variants that we don't cover in our catalogue, we have a really experienced and excellent custom design team who can make you something with the specialist requirements that you may need. So yes, changes to the allele frequency and also to the variants included in the reference standards is possible, and we do process a lot of projects like that day to day.
Oh wow, so customization to the max. That's really cool.
Yeah.
That is awesome. All right. Oh, cool. So there's a few more questions that are coming in. Let me check the time. Oh, we're doing good. Okay. So here's another question. It's, is it possible to extract both DNA and RNA from the RNA reference standards?
Yeah, another good question. We have had some interest in that in the past. The RNA reference standards are sold as an FFPE curl, including the cell lines that contain those fusions. So ALK, RET, ROS1, NTRK1 and NTRK3. We only validate the reference standard and those fusions at the RNA level. However, there's no reason that customers couldn't also extract the DNA from the curl since the cell lines are there. And we actually did a technical study on this and have an app note available on our website describing how customers could perform dual extraction of both DNA and RNA from the RNA reference standard curls if they desire to do that. So have a read of the app note. I think we recommend which extraction kit we used, but keep in mind that we only validate the fusions at the level of RNA so far.
Okay, good to know. Okay, so it does look like we're approaching the end of our time slot, but we've got time for just this very last question. Let's see. It says, do you have any reference standards for microsatellite instability?
Yeah, we're seeing microsatellite instability as a growing area, and we've had a lot of requests from our customers for this. I think we're seeing increased testing and an increased need for a reference standard there. Currently, we don't have one available in our catalogue, but we're planning to launch one at the beginning of next year. So it's quite exciting. It's one of our most interesting R&D projects at the moment. So we do have some material that we're testing in-house, and hopefully that new MSI reference standard will be coming to market early next year.
Wow, exciting times in the field of OncoScan. Wow. Hannah, thank you so much for being here and sharing this great information with us. And for the audience, thank you so much for your participation. You guys are really what makes this webinar so interactive and so fun for all of us. And thank you again just for all your questions. I see that there are a few more questions remaining in the queue. If your questions were not answered, one of our scientists will respond to you via email. If you have further questions that did not get submitted during the broadcast, please email them to info@horizondiscovery.com. All right, before we go, we would like to thank both LabRoots and Horizon Discovery for underwriting today's educational webcast. This webcast can be viewed on demand through January 2019. LabRoots will alert you via email when it's available to replay on demand. We encourage you to share this email with your colleagues who may have not been able to attend today's live event. An exit poll will come up on your screen. Please, 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 please share your feedback with us. It'll help us plan for all future webinars. Until next time, we'll see you soon. Goodbye.